All right, yeah, got it. Too.
Okay, all right. Well, we've got that type of stuff. Okay. Now the next thing we've got, the different topic. We have overviewed the major processes of RNA processing. For things like the future messenger RNAs, you have the splicing and alternate splicing that allows one gene to code for many different potential messenger RNAs. We have the capping, the addition at the 5' or front end of the RNA of a modified, in this case, a 7-methylguanosine for the ones that are messenger RNAs. That cap is absolutely essential for ribosomes to recognize and bind to it. Without that cap, the ribosomes don't know it's there, and therefore translation does not occur. We'll see that's actually used as a regulatory mechanism on translation in some cases. Okay, so you have it. There are other caps that other types of messenger RNAs have as well. There's one group. In fact, it's the small nuclear RNAs that make up major components of splices. So they have a trimethylguanosine cap. In other words, guanine with 3 methyl groups on it. And that's unique to these types of RNAs. And other RNAs may have other. So the caps are often used as tags telling the cell what kind of RNA they are and what to do with them. And then, unique to messenger RNAs, we have the poly A tail, a stretch of about 200 or 300 adenosines on there. And that plays a role in regulating the lifespan of the messenger RNA, and also it enhances. It's not essential for ribosome binding, but it ups it quite a bit. Okay, so it helps in translation, and it also helps protect the RNA. And there are sequences between the so-called untranslated regions. More of that when we talk about translation. Okay, now, other kinds of RNAs, for instance, if you look at the ribosomal RNAs, turns out that those are processed differently. 3 of the 4 ribosomal RNAs, what we call the 28S, that's the one that's the ribozyme that actually catalyzes peptide bond formation in ribosomes. In other words, it actually joins amino acids together. It's an RNA enzyme or ribozyme. The 18S, which is part of the small subunit, the 5.8S. All 3 of those RNAs, and there's a 4th one called the 5S, but all 3 of those RNAs are coded for by a single gene. So you get a gigantic precursor RNA, and then enzymes, including certain SNRPs like U3, will join into a complex and cut that big precursor RNA into one piece, the 28S, another piece, the 18S, and a third piece, the 5.8S, and throw other portions out. And then they get modified too. So that has a different processing pattern. Each of these RNAs are going to have certain kinds of processing before they're finally a mature functional messenger RNA. As we'll see later, these things called microRNAs and small interfering RNAs. The RNA precursors are cut in a specific way by an enzyme complex appropriately called dicers. It slices and dices. Okay, and then that's the mature RNA that gets transported inside the cell. So the key thing is here, folks, RNAs have to be properly processed before they go into the cytoplasm. Okay, so only processed RNAs, fully, completely processed RNAs can go into the cytoplasm. What we got here?
She was having trouble reading.
I know. Alien script. One of those newly discovered exoplanets, they come from there.
Trying to like go for my
Okay, yeah, all right.
I have no idea.
Well, hey, just think, if you join the military, you ought to have experience in code breaking, right?
Oh, yes.
Yeah.
I can put that
If
on
you can
my
read,
resume.
if you can read that, so yeah, I read Sauterer's lecture notes, and I could read that, I could break any damn code in the business. Okay, well anyway, unprocessed RNA cannot leave the nucleus. So that allows us to do things like, one, you don't want, for instance, you would not want a partially processed messenger RNA containing things like intron sequences, stuff like that, to go in the to go in the cytoplasm, be grabbed and read by a ribosome, and putting in amino acids you don't want because it's reading an intron and it doesn't know it's doing that. Okay, that would be very, very bad. You don't want a huge RNA that's not properly processed to go into the cytoplasm. God knows what it might do. At most, at best, it's a waste of resources. At worst, it can cause downright harmful effects in the cell. If you accidentally screw up and mistranscribe a gene, that happens all the time. Recall, we saw that the majority of genes, that the vast majority of genes, you would see in the nucleus low levels of transcription, not much, but it was there. And if you're transcribing a gene that codes for like a master control transcription factor, for instance, the ILIS/PAC6 gene, you don't want that gene being expressed in cells that you don't want. You don't want eyes growing on your knees unless you got some crazy molecular biologists who are deliberately trying to do that, right? Or literally eyes in the back of your head. That can happen. Okay, so you don't want these genes expressed in the wrong places. So if you mistranscribe a gene, you got a chance of undoing your mistake because you won't process that nuclear RNA and it stays in the nucleus unknown to the cytoplasm and eventually gets degraded. So the question is, how does the nucleus and whatever gets things out of the nucleus, how does that nuclear transport machinery know that an RNA is fully processed or that is not fully processed? We have to have some kind of recognition mechanism to know what RNAs are processed and what ones aren't. And the ones that are unprocessed stay in the nucleus until they get processed or until they get destroyed. The ones that are processed get out. So use the analogy of like going to a concert or rock concert or a football game or something like that. They're not going to let you in unless you have a ticket, or maybe you know the gatekeeper really well, okay? So you have to present this ticket to the gatekeeper. Say, oh, this is a legitimate ticket, come on in. If you don't have the right ticket, or you have a ticket to another concert, and he looks at it, Muslims probably don't, and he looks at it, this is not the right ticket, it's for last week's concert, you just say, get out of here, you know. That kind of stuff. They're not going to let you in, okay? So we have, because they're looking at this ticket, that's the mechanism saying that you have bought the ticket and paid these guys a whole lot, far too much money to keep them nice and rich, and they let you in based on that. So in a sense, we must have to have some kind of ticket on processed RNAs and stuff. And we have to know what the gatekeepers are. And we'll see in the most part for nuclear and cytoplasmic transport, we'll go in more detail when we come back on Thursday. The nucleus, of course, has a double membrane, not just one membrane, two membranes. The membranes are distinctly different from each other. The outer membrane is actually continuous with the endoplasmic reticulum membrane. It has a different composition, proteins and lipids. The inner membrane is completely different. So you have two membranes. Underneath the inner membrane, you've got a substructure called the nuclear lamina that acts like a supporting framework. Plays other important roles too. More on that later. Okay, so you have the nucleus, you have to not only get something through one membrane, you have to get through two membranes. And you have to get it through this filamentous layer beneath the second membrane. That's no problem, it's very porous. Okay, but you got to get through two membranes. And in these membranes, there are numerous, we could call them gatekeepers, just like you go to a stadium. And there are going to be a number of gates where people can get into the stadium or out of the stadium. You can't get in, unless you're really good at climbing, you can't get in the stadium from areas outside the gate, unless you parachute in and you're an expert climber, right? And the cops don't see you. So you only get in or out of the stadium through the gates. Same thing, the nucleus, you have these gates. And these gates are called the nuclear, and we'll go through these guys later, pore complexes. They're often called nuclear pores, but I don't like that term because it just sounds like it's a hole in the nuclear membrane. It's not. It's a very complex, specific structure. We call them nuclear pore complexes, or NPCs as another abbreviation. Those are the gatekeepers, and we're going to cover those guys later on. But the key thing is, how do we get these things in and out of the nucleus. Okay, these gatekeepers have to recognize a ticket saying, I am fully processed. And if they don't have that ticket, they will not transport you out of the nucleus. Now, by the way, the nuclear pore complexes are involved in transporting things from the cytoplasm into the nucleus. So you have to have entry tickets to get into the nucleus, and you have to have exit tickets to get out of the nucleus. And the tickets are going to be different. If you had the same ticket, you want to know what direction you're supposed to go, right? And the gatekeepers want to know what. Do you go in or do you go out? Okay, so you have to have two separate tickets. An entrance ticket and an exit ticket. Okay, well, what are these things? And how does this happen? Well, what it is proteins that bind to completely processed RNAs, but don't bind to ones that aren't fully processed. Okay. Okay, now, when we talk about RNA, your typical molecular biology course, you see RNA and people draw as a nice, nucleic acid, right? But in reality, RNAs are not found, at least in eukaryotic cells, as just strings of a nucleic acid. Instead, RNAs are always complexed with various RNA binding proteins. So you never have naked RNA any more than I'm going to be walking naked. You're going to have a naked professor in this class. It just ain't going to happen. But you probably won't want to see it. I'm surprised my wife wants to see that. But okay, but anyway. Okay, so I'm always wearing clothes. Okay, now on the other hand, so all these RNAs are going to be complex with proteins. You have an RNA and you have a whole bunch of different proteins that are sticking to these RNAs. Some stick all over the place, some are sequence specific. And this whole complex RNA protein complexes are called RNP. So instead of a messenger RNA, we could call a messenger RNP. Okay, now there's a bunch of different proteins. There are common proteins that you find. There are sets of proteins that bind to nuclear RNAs. And we often call them HN, heterogeneous nuclear, RNP proteins. Because they bind to the nuclear RNAs. Many of them are common. When you look at any nuclear RNA, at least if you're looking at pre-messenger RNAs, and you'll find a constant set of these HNRNP proteins, and they've identified and characterized a number. But you also find that there are ones that are unique to specific RNAs. So we have common and unique. In other words, RNA specific, I would call it specific, HNRNP proteins. Okay, now once you go out of the nucleus and into the cytoplasm, these HNRNP proteins start getting replaced by a different set of cytoplasmic proteins, both common and specific. And we have the messenger mRNP proteins. And once again, there are ones that we find in all of them, or almost all of them, and we have specific versions. Okay. That's sort of the outline of these kinds of things. And it's the proteins that for the most part, now RNA sequences may play some roles in recognition by the nuclear pores. In other words, play a role as part of the ticket. But for the most part, the entry and exit tickets have something to do with these associated proteins. Generally, that's what the nuclear pore complexes recognize. Like I said, they may recognize RNA sequences in some cases, but for the most part, proteins are bound to these things. Now let's take a look at an example here. Let's take a look at a mature messenger RNA. We've processed it fully. We've got our cap here. We've got exons but no intron sequences. We have the stretch called a 3' untranslated region, more on this later. And that's a region of a few hundred nucleotides before you get to the poly A tail. Let's look at the nuclear RNA. What we have here is we have a cap site but no cap. We have introns and exons. And we have a polyadenylation site but no poly A tail, and then finally stretch left. So what we see here, what's the new things we have in this messenger RNA? Well, number one, no exons. No introns. Unless you deliberately throw a little intron sequence in from alternate splicing. But for the most part, we've gotten rid of the introns. That's one difference that can be recognized by binding different sets of proteins. Second thing, we have a 5' cap, this 7-methylguanosine. That could bind proteins that play a role as being a ticket to the cytoplasm. Third, we have that poly A tail. That could provide and bind proteins that act as your ticket to freedom and out of the nucleus. So we'll put some proteins over here. And we'll call them export proteins, or say that they have what we call export sequences. Tickets that get you free from the nuclear prison. Out here, we could also have export proteins, or proteins with export sequences. If this protein binds only to a cap, it will not bind to the cap site. That's not a cap, that's just a recognition site for the enzyme that puts the cap there. If we have proteins that bind to the poly A tail, well, there ain't no poly A tail here. There's a site that tells the poly A enzyme complexes that make the poly A tail. There's a site that'll bind these things, but that's not poly A tail. These export proteins require the poly A tail here. So here, in this case, we've got export proteins here and here. Now, by the way, it turns out that you don't have to have, for messenger RNAs, you don't have to have a fully completed 7-methylguanosine cap in order to get out of the nucleus. It helps. But some messenger RNAs are put out of the nucleus with a guanosine, but not a 7-methylguanosine cap. That will allow it to get out of the nucleus, but lacking the 7-methylguanosine cap, the ribosomes will recognize it. That means that messenger RNA is in the cytoplasm but cannot be translated. Later on, you'll stick the methyl group in the appropriate place, and now the ribosomes can read it. And we'll see, especially when we look at oocytes, it's one way of storing messenger RNAs in the cytoplasm for long periods of time, but not translating them until we really want to do so. That's coming up later. Okay, so we've got these export proteins that bind to sequences that are uniquely added to mature messenger RNAs that are not found in the precursors. Now this is a little more, I don't know for sure on this, but a little more speculation on my part. Suppose we have some proteins that would bind to certain things in intron sequences. I'm going to put this in a question mark. Do we have proteins that act as export inhibitors? They're present on sequences that we splice out and discard? Maybe. I don't know of any, for certain of any examples of that, but I certainly am not going to rule it out. That could provide us additional level of control. That sequences that you're supposed to get rid of, but in order to process the RNA, but have not gotten rid of them yet, would have things that would actually act to prevent export. Kind of like the stripes on the suit in the guy in jail, right? The guards won't let the guy out. If you're wearing prison stripes, they probably won't let you out unless they're really certain that these are not really supposed to be on you. Okay, so we have certainly export proteins that recognize and bind to these sequences that are unique to processed RNAs. We may well have export inhibitors that bind to sequences that are supposed to get cut out or modified or otherwise removed. In these cases, that makes sure that only the properly processed RNAs get out of the nucleus. If we mistranscribe a messenger, a particular gene, we have the precursor RNA. We may have export inhibitors attached. What we are missing are the appropriate splicing factors that allow that nuclear RNA to get processed into a messenger RNA. We're in the wrong cell type. We made a mistake here. We don't have the splicing factors, so we never go from here to here. Therefore, we never get the export proteins binding to where they're supposed to bind because those sequences aren't there. End result, folks, those RNAs stay stuck forever in the nucleus till death do them part. And eventually that's what happens, nucleases get. Okay, so that's the basic idea of how we recognize and get things out of the nucleus. Now what we're going to look at when we come back on Thursday is we're going to look and see how the export machinery in the nuclear pores actually work. And then we'll be in the cytoplasm and we'll start looking at all kinds of cool stuff in the cytoplasm. Save time, let's see, just the list. Okay.